grinding of ultrasonic vibration tannins extraction from oak bark. Oh, it's very interesting. Are you ready, Maria? Please.
Yes. Yes. Good morning for everyone.
Your language of your report, English?
In English, yes.
Okay, right. You can start, please.
Okay. So I want to present.
Sorry, just a little bit. Yes, right. That's a good vision.
Good. Yes. Okay.
Yes.
So I want to prepare our presentation, and topic is an influence of grinding, shaking, and ultrasonic vibration of tannin extraction from oak bark. And first part is actuality. I'm sorry, I'm a little nervous because it's my first time when I speak on this conversation. Tannins are the class of polyphenolic plants biocultures which have an androgen effect, and their biological activity is based on the ability to bind and precipitate proteins or other organic compounds, including alkaloids and amino acids. Tannins are widely distributed in many species of plants, but in this work with oak bark is used as a source of proteins precipitating tannins. Plants use tannins for protection against bacteria, fungi, and some insects. Tannins are mainly used by industry from leather processing. However, recently, antiviral activity of tannins has been discovered. Tannins showed antiviral activity against human immune viruses and herpes simplex virus 2. In case of herpes simplex virus 2, silver nanoparticles with tanninic acid act as a microbicide by providing a supporting of viral protectile by the cell. And in relation to HIV infection, the killing substances inhibit the replication of HIV-1. Science all that tanninic substances used in the experiment significantly inhibited the production of the P24 antigen. Therefore, tannins, in addition to their biological role, also play a very important role in the industrial sector, animal feed, meaning chemical and tanninic industries. So we considered work to finding ways to better extraction. Okay, aim and task of work. The main aim of the work is to evaluate the influence of various factors on the extraction of tannins from the oak bark. And tannins were extracted with 50% water ethanol solution for 20 minutes. We have applied the combination of treatment of extraction, efficiency improvement, a shaking of barks with extract on laboratory shaker SHO10 during extraction, and sonication of barks with extract on laboratory ultrasonic bus IM4 during extraction, and the grinding of oak bark to precipitate of size 0.8 millimeters with the help of laboratory grinding machine MLT2. And non-ground bark was used as a control. On this picture, you can see a 4 petri dish with agar gel, but with some special ingredient, and this is albumin. If I remember correctly, we use bovine albumin, and albumin is a family of globular proteins, the most common of which are the serum albumins. Stage of work. First one is the precipitation sample. We had grinded part of our bark in a grinding machine. It's like little meal in laboratory, and then we use sorry. And then we use ethanol solution to start off extraction. After 20 minutes of extraction, we started at centrifugation. Why we don't use filter and use centrifuge? It is because we use this for particle deposition. Our parts of oak bark was so small. And at the end of our work, we did a radial diffusion test. And this is our result, radial diffusion test. And you can see on this picture, this white circle, it's our result. Because if you remember that we use the albumin such a source of protein, and tannins precipitate it. Also, we did this table. As a result, we had to measure 3 diameters of this white rings and have average mean for 4 samples: shaken of ground samples, shaken of non-ground samples, sonic of ground samples, and sonic of non-ground samples. And then we did a statistical analysis of it. And the performance project has 4 parts. In each part, the null hypothesis was proposed that there was no difference between the 2 means of precipitate rings diameters. The t-test was used for testing the null hypothesis in Excel. In the first part of the project, we compared tannins content in the shaken extract from ground and non-ground oak bark samples, and the value of t-test obtained was 4.79, and the probability of obtaining this value for a 2-tiled test was 0.28 multiply 10 in power minus 3. So we are able to reject the null hypothesis and conclude that there is significant difference between tannins content in the extract obtained with the shaking of ground and non-ground oak bark samples. In the second part of the project, we compared tannins. I'm sorry. 1 second. In the second part of the project, we compared tannins content in the silicate extract from ground and non-ground oak bark samples, and the value of t-test obtained was 1.74, and the probability of obtaining this value for a 2-tiled test was 0.134. So we are able to accept the null hypothesis and conclude that there is no significant difference between tannin content in extract obtained with a sonication of ground and non-ground oak bark sample. In the third part of the project, we compared tannin content in the shaken and sonicated extracts from ground oak bark, and the value of t-test obtained was 3.95, and the probability of obtaining this value of 2-tailed test was 0.0014. So we are able to reject the null hypothesis and conclude there is a significant difference between tannins content in extract from ground oak bark obtained with shaking and sonification treatment. And the last part of the project, we compared tannin content in shaken and containing extracts from non-ground or bark, and the value of t-test obtained was 9.28, and the probability of obtaining this value of 2-tiled test was 0.23 multiplied 10 in power minus 6. So we able to reject the null hypothesis and conclude that there is a significant difference between tannins content in extract from non-ground oak bark obtained with shaking and ultrasonication treatment. And our conclusion was that as a result, we propose to use grinding and shaking for arc bark in order to improve tannin extraction efficiently. And experimental result was that ultrasound treatment has no statistically significant effect on tannin extraction for oak bark. Thanks for your attention.
Okay, thank you very much, Maria. Have you any questions, colleagues? Please, you can ask Russian, you can ask on any language, I hope. Yes, please, Olena Alexandrovna, please.
Yes, thank you, Maria, for presentation. It was very important. And I have a question. So you mentioned that the concentration of water as no solution was 50%. Why did you select this value of concentration? And what about other concentrations? How other concentrations may affect the tannin content in the extract? Did you investigate such issues?
Окей, чому ми використовували саме 50% розчин спирту, правильно?
Так.
Марія, ти, можливо, було відкрити презентацію, можливо, вона б ще хтось би задав би по слайдам. Не треба закривати ніколи раніше.
Добре.
Окей. Yes, sorry, please, answer question.
Окей. Якщо я правильно пам'ятаю, ми використовували такий розчин етанолу з водою, тому що 96% розчин спирту, він, по-перше, буде швидше вивітрюватися.
Ні, Марія, згадайте, будь ласка, ми говорили, що є таніни водорозчинні, а є таніни спирторозчинні. І тому ми хотіли спробувати і ті.
Так.
Отак.
Горілці найкраще, мабуть.
Так.
40%.
Так.
Водорозчинна фракція, спирторозчинна фракція. Окей, пліз, Тетяна Володимирівна, Олена Олександровна, вибачте, все нормально? Відповіли вам на запитання?
Так, я отримала відповідь.
Дякую. Тетяна Володимирівна, ви ще піднімали руку, здається.
Так, я хочу спитати, скажіть, будь ласка, яка була повторність ваших дослідів? І хто взагалі вивчав таніни? Ви можете нам сказати?
І на чому вивчали?
І на чому, так. От. І які ще рослини чи живі об'єкти є джерелом танінів?
Так, джерелом танінів є більшість, скажімо так, фруктів, які проявляють в'яжучі особливості. Я точно пам'ятаю, що гранат містить в собі дубильні речовини і зелений чай. А також із продуктів більшість вин.
А там питання було, як ви визначали і на чому, можливо, чи визначали взагалі ви відсоток танінів? Саме таніни ідентифікували ви чи ні? Питання
І
було,
повторність дослідів.
І повторність, так? Да?
Угу.
Що скажете, Марія?
Так, ну, як... Ми не визначали вміст танінів, тому що ми знали, що в корі червоного дуба вони є. І якби суть нашого експерименту полягала не в визначенні танінів, а в покращенні їх екстракції. Ось,
а як ви могли, не визначаючи танінів, оцінювати покращення екстракції, якщо ви їх не визначали?
Тобто, це було вже відомо про наявність танінів.
Ні, це не факт.
Ні, там по-іншому.
Там
відбувається реакція преципітації. Діаметр кільця преципітації залежить від концентрації танінів в екстрактах. Тобто це відомо, це з літератури відомо. Ми ставимо реакцію преципітації альбуміна в гелі і далі вимірюємо діаметр кільця преципітації. Щодо повторності, ми робили 8 повторів кожної комбінації факторів.
Добре, дякуємо. Взагалі, зрозуміло все, питань немає. Це не дискусійна, це у нас конференція. Окей, чи має ще хтось запитання, будь ласка, до Марії?
Можна запитати.
Да, будь ласка, питайте всі. Шановні студенти, підключайтесь активно, це ваші колеги доповідають, так що питайте.
Так, в мене є питання. Дивіться, а як ви з дубової кори танін, екстракт витягували, і чи ви використовували вже готовий промисловий? І також, я розумію, це реакція інвітро, так? І саме проти яких вірусів або з якими культурами ви працювали? Чи це було чи це був біологічний матеріал, чи це була сам модель як
Можна я роз'ясню, можливо, Ангеліна не почула, вони там проблеми були з підключенням. Вони ж пояснювали, що не екстрагували таніни, а була реакція по реакції преципітації перше. А які види, які містять таніни, Марія вже, по-моєму, досить, щоб не повторювала, не мучити її, вона вже, по-моєму, це розказувала. Які об'єкти біологічні рослини містять таніни. Чи задовольняє вас такий варіант? Чи, можливо, Анна Вікторівна ще додавить щось?
Я хотіла б додати, що ми не тестували біологічну активність, тобто ми не працюємо з вірусами в лабораторії. Ми могли б зробити антибактеріальну активність, але це наші майбутні плани. Тобто це відомо з літератури. А ми перевіряли саме методи обробки або ультразвук, або перемішування, або подрібнення, наскільки вони будуть впливати на вміст танінів в екстрактах.
Тобто фізично, наскільки можна видавити їх з допомогою додаткової вібрації, фактично.
Так, так.
Тому що ультразвук, до речі, sorry, I have a question. Just it's arised from your answer. What type of activity, of ultrasonic activity, I mean? You know, there's any limit of cavitation limit, yes. What type, на який рівень ви використовували ультразвукової обробки? Що це було? Тому що є кавітаційний ефект, де можна вибивати все, що завгодно, за рахунок розруху, руйнування клітин. А які ви використовували?
So we use laboratory ultrasonic bath. Very powerful. It has 3 sources of ultrasound. And we have only the model of this bath, and we can use some passport and see the maybe some parameters, but we don't change such parameters of the power of vibrations. So, and we know that this bath creates cavitation effect.
Yeah, but what type of amplitude, or technical parameters?
We don't have such parameters.
It's important, I hope, because it should be used for any effective result. Maybe it's more used for, because I know that's just one time I deal with ultrasonic effect and like a transformation effect, but it's a specific type of. So sorry, that's another question.
Thank you.
Have you any other questions, colleagues? No, thank you.
Извините, но я не поняла так про культуру самого вируса.
Нема
Вы работаете с готовой прощой?
Ангеліна, вибачте, щоб уже скоротити, вони працюють з корою дуба. Вони сказали, ніякої вірусів нема. Вони визначали лише можливість екстракції, непрямий показник там по преципітації. Все, ніяких культур вірусів, бактерій поки що не працювали вони. Я правильно відповів?
Так, а смисл концентрации танина, если против чего не був використаний? Просто прямий метод?
Наступний етап. Це просто поки що, ви знаєте, що будь-яка методика степ-бай-степ іде. Це методика екстракції. А далі методика використання буде наступний. Очистки, використання і так далі. Якщо правильно я вас зрозумів, Анна Вікторівна.
Ми хотіли підвищити рівень екстракції.
Так, це головне.
Більше танінів виділити з одиниці маси.
Антибіотикотерапія. Смисл тоді
Ні. Це наступний етап, Ангеліна. Кожне дослідження розбивається на етапи. Перший етап – це екстракція, а далі вивчення вже. Порадьтеся, сконсультуйтеся зі своїм керівником.
Просто выделение, экстракция и метод обычный, это просто выделение метод был или как?
Так, це метод екстракції, метод виділення означає.
Удосконалення методу, удосконалення методики виділення.
Досконалення.
Ну, понятно. Спасибо.
Окей, шановні колеги, чи є ще запитання до Марії, до її доповіді? Марії Юріївни? Немає. Дякую, Марія, пані Марія. Наступне у нас, згідно нашого плану, це доповідь. Зараз подивимось, хто там буде читати у нас. Я не знаю, хто там буде доповідати, але це доповідь Sentence gum as a component of starch-based bioplastic. Тертичний Кайданюк. Хто буде? Кайданюк?
Так,
Денис.
доброго дня всім. Ми будемо презентувати разом.
Так, це наші студенти, які теж розвивають нові підходи. Будь ласка, хто буде? Денис доповідати, так? Чи Максим?
Першу частину буде доповідати Максим, а пізніше я.
Он як. Окей, давайте тоді. Але довго не розтягуйте, шановні дорогі, бо ми так вийшли, вже вибилися з графіку. Я прошу, будь ласка. Окей, починайте.
So the topic of our presentation is xanthan gum as a component of starch-based bioplastic. Next slide, please. So mankind uses plastic everywhere, so the production capacity of this important material is only growing every year. There is an environmental risks that are based on huge amount of plastic waste which are not disposed of but only released into the environment. So alternative sources of this material have been developed for some time, which will be more environmentally friendly. In fact, the use of starch in this industry has been going on for a long time. For example, this component is added to existing plastic formulations to improve performance or reduce toxicity. Therefore, this investigation was performed to demonstration of production starch-based bioplastic with xanthan gum in laboratory scale. The object of research was the starch-based bioplastic with xanthan gum. The subject was to find indicators of flexibility and resistance to mechanical action. The purpose of work to improve the created formulation of starch-based bioplastic due to addition of xanthan gum. The main task was to prepare the new formulation of bioplastics with different proportions of xanthan to starch, then to investigate influences of addition of xanthan gum to formulations of starch bioplastic, and to investigate the application of thermostat for acceleration of drying the bioplastic. During this investigation, we used previously created formulations of bioplastic from the starch and replaced some amount of starch by less amount of xanthan gum. The main purpose of xanthan gum application was its thickness, stabilizing, suspending, and emulsifying properties. Xanthan gum is high molecular weight polysaccharide produced by the bacterium Xanthomas campestris. It may be degraded, but it's not readily degradable. But for application in relatively small quantities, it can be harmful and can degrade during some time. Starch is used as main component due to its thickness, water absorption, and strange potential which it can provide for bioplastic. Glycerin is used as an additional plastizer which interacts well with the polymer starch. It increases the elasticity of the material and prevents its destruction by reducing its fragility. Vinegar is used in the formula because it accelerates the hydrolysis of the bonds of the starch components, which helps the molecules to form stronger bonds. And water is present in the recipe as the main binder. It also has a positive effect on elasticity of combination with glycerin. So here you can see the prepared formulations of our samples. The only difference between them is amount of starch and amount of xanthan gum due to one another. And here you can see the experiment processing scheme. So in the first step, we mixed water with starch and xanthan gum. Then we added vinegar and glycerin. After that, we put the this mixture to the water bath at 85 to 95 degrees and mixing it for 10 or 15 minutes. Then we distribute the product in the flat surfaces. After that, we put that surfaces into the thermostat for 4 days at 50 degrees, and then we obtain the final product. In this photo, you can see the samples before the when we just mix it. Then you can see the process of heating in the water bath. Then you can see in figures photo 6, you can see distributed samples on Petri dishes, and then you can see in picture 7, it is a thermostat that our cafedra is equipped with, that we use to dry our samples. So next continue my товарищ.
So now I let continue, and the next stage, the results of experimental part. There you can see they are obtained by plastics, and the control variant is 10 grams of starch only without the xanthan gum, and the best variant, as you can see, with 0.5 gram of xanthan gum and 9.5 gram of starch. Also, there are such variants with other formulations, and you can see their appearances. And there you can see the table with main characteristics of prepared formulations. The main differences here in amounts of xanthan gum and starch, they have characteristics of flexibility and strength, and they are ready for use or not. And as you can see, the control variant from our previous investigation is ready for use, flexible and strong. And in comparing to it, our new formulation with 0.5 gram of xanthan gum and 9.5 gram of starch is more flexible and strongest than control one. And also the other formulations are not so strong or flexible. One of them with 13 grams of starch and 2 grams of xanthan gum can be used for limited use, for limited application. And there, for example, you can see. There you can see the examples of application, the control one, the formulation in 0.5 gram of xanthan gum, and their variant with 2 grams of xanthan gum and 4 grams of starch. And there you can see that they are flexible, they have a good thickness and strength, and also such bioplastics can be used for production of current films, wall-smoked bioplastic items, and therefore can be used for cosmetic, food, chemistry, agriculture industries, and so on. Also, as a part of our input, is investigation of biodegradation of experimental bioplastics. And in previous input about the control one, also was investigated biodegradation, and it was summarized that after 3 days of being in water and seawater and soil, the bioplastic lose its properties, the flexibility and strength, and lose up to 50% of volume. The bioplastic with xanthan gum requires more time for biodegradation because after 3 days of samples lose just from 35 to 40% of volume, as you can see there. Also, it's necessary to know that first of all, samples lose their thickness and become more transparent with time. And for example, under boiled water at 100 degrees of Celsius, samples dissolve after less than 1 minute or even less than 30 seconds. And there you can see the examples, the best formulations with xanthan gum. At the left side it's still water, and right side is seawater, and you can see. And prospects for development, for decreasing the prime cost of starch there can be used alternative cheaper raw materials instead of corn or potato that is convenient for production of starch. Yeah, and for its production, there are such raw materials as seeds, fruits, and their waste can be used, and we will consider it in the next slide. Also, the xanthan gum is a relatively expensive constituent, of course, but laboratory scale production, because it required small amount of xanthan gum, of it using xanthomonas campestris as a part of whole production scheme, can decrease prime cost of bioplastics. Also, it's possible to improve the properties of the final product by mixing during preparation by special mechanical mixers for better homogenization, because we use sticks and mix by our hands, yeah, and it's possible to create samples with specialized shapes for special usage, the shapes of plates, shapes of cups, pans, etc., for different purposes, and it's required to more investigate the biodegradation properties of xanthan gum, as it was said earlier. So, as I said, there you can see the alternative raw materials for production of starch, the method of extraction, and starch yield and purity percentages. They can see that from scientific research, as is referenced in our work here, there can be used mango kernels, pineapple stems, jackfruit seeds by 3 different methods of starch extraction, yeah, as you can see, and banana peel. And as you can see, to the according to the starch yield, the jackfruit seeds obtained by method of distilled water by extraction, starch extraction method, has the best starch yield, 84.48%, and there are good purity, starch purity of all examples except one with using of banana peel. Yeah, that's just 70% of starch purity. And also you can see that banana peel, pineapple stems have not so good starch yield, but also can be used, because that's first of all saying about application of waste from production, yeah, from industries. So, conclusions. In this work, we investigated the production of starch-based bioplastic with xanthan gum. Its main advantage is biosafety. The small amount of xanthan gum is not toxic for environment, and such product start actively biodegrade after 24 hours. The addition of xanthan gum positively affected functional properties of final product, the flexibility and strength first of all. The application of thermostation at 50 degrees of Celsius for acceleration of drying time could reduce the time of drying by 2 times, from 7-8 days to 3-4 days at thermostat. Of course, it's too early to talk about the commercially ready product for global usage, but the potential prospects for improvement to today's formulations, the properties, conditions for industrial production. It's important to know that these formulations are ready for use as current films in cosmetic, food, agricultural industries just now. Thank you for your attention. That's all. And there also you can see the references.
Okay, thank you. Thank you, colleagues, young colleagues. So have you, dear colleagues, have you any questions to our young colleagues? Please ask them. Have you any questions? Yeah, please, Mikhailo Ivantsov, please.
Thank you for such amazing report. I have a question. You mentioned that you can change the materials in order to increase starch content, but can you, for example, tell whether changing of materials can change the speed of formation of this starch?
Yeah, I can note that it was said that the application of alternative raw materials for starch production is used not for increasing the starch content. It's used for substitution the convenient raw materials as starch, as potato, as corn, because they used for food industry, yeah, and it's obtaining the starch from such waste, for example, not from traditional convenient potato corn, is more як це сталий українською мовою, тобто stable, yeah. It's more about the biosafety, the eco activism and so on.
So, as far as I understand, your aim was not to create the speediest way to formate this bioplastic, but to make it like reusable, that you like put in garbage your trash and then you like remake plastic again and again and again. So to make more copies of it.
Yeah, the use of such raw materials is not for acceleration the speed, not for increasing the starch content. It's for substitution there, convenient raw materials, and the main purpose is to create the bioplastic, biodegradable bioplastic from starch with xanthan gum addition, which can be used for, as I said, different industries to substitute their convenient plastics from petroleum, yeah, and so on, to decrease their environmental problems caused by plastic pollution and so on.
Thank you for the answer.
Yeah, thank you. Have you any other question, please, colleagues? Have you? No, just let me ask you, or just as I understood, this problem has two sides. First, any requirements to physical characteristics, I mean strength, stability, or something like that, yeah?
Yeah.
And then another specificity of biodegradation, of destruction of the plastics, of bioplastics, that should be prevent any additional contamination of ecological systems, right? So just to the physical characteristics, I have no questions because that may be modified using any other additions, additives, and so on. But my question is to